medicine gastroenterologist. He trained at the University of Minnesota and completed his fellowship in gastroenterology here. He also trained in basic immunology as a Howard Hughes Physician Fellow under the mentorship of Dr. Mark Jenkins in microbiology. He's going to talk today about the human microbiome. Dr. Khoruts.
Thank you. It's a little premature. All right. Try to orient a little bit. So my purpose is, it could be a fairly intense talk, but the messages are fairly simple. And I just wanted to introduce the concept that we have a true organ made of microbes, which is a little bit of a paradigm shift, which creates discomfort with patients, physicians, regulatory agencies, etc. But our notion of microbes has been shaped by infectious disease, which is a bit of a different paradigm than the one I'm talking about. And these were, in Koch's postulates, ideas, single pathogen, single microbe, and whatever disease it is. And of course, our human history has had a lot of tragic interactions with microbes. Some of the illustrations here are images of Black Plague, the essential extermination of Native Americans by smallpox, which allowed colonization of this continent. And multiple others, as well as some key players like Pasteur and Koch that are thrown into this picture. But of course, we have a lot of microbes in our own bodies, and as you've all undoubtedly heard, there's more microbes living with us than the rest of the cells. Most of them, by their sheer biomass, live in the distal gut, but they, of course, they're present in all surfaces. And if you just look at the sheer biomass of us, considering the shedding and turnover of microbes, there's actually more microbes than the rest of us. We produce about 5 elephants' worth of fecal material over a lifetime. So by sheer biomass, we're about 10 to 1 microbe over a lifetime. And certainly these microbes have their own destiny and purpose, and they play important roles in physiology, which is something I'll talk about. It's a very diverse world from some perspectives. For example, all domains of life are present within gut microbiota, the domains of life being bacteria, eukaryotes, archaea. There's viruses, mostly bacteriophage. But at another level, it's a highly selected group of microbes, and the number of phyla, for example, living in the distal gut is dominated by the number of microbe phyla is dominated by just 2, Firmicutes and Bacteroidetes, while in soil you'll find up to 100 different phylum. And a glimpse of what the microbes do can be obtained from just looking at germ-free animals. We have a number of abnormalities. Obviously, if you take care of these animals, you have to attend to their specific nutritional needs. There's certain vitamins that we can't make that you have to supplement. But they are also smaller animals. They're about 40% of the body weight of the wild-type mice. And along with that, they have less body fat, smaller heart output, etc. They have decreased metabolic rate. They have somewhat different behavior. They actually move around. They actually eat more despite their lower body weight, but they move less. Their intestinal motility is altered. It's slower, etc. And of course, they have great susceptibility to infectious disease. Just the number of infectious organisms that it takes to infect a germ-free mouse, not surprisingly, is orders of magnitude smaller. This just shows just looking at these germ-free animals, they are smaller in weight, they have less fat, and yet they eat more chow. This is looking at what happens when you conventionalize them. And actually that was present on this figure as well. They gain weight. And the normal phenotype is restored by just transferring bacteria into germ-free animals. If you look what happens to their intestines, they may have at first shorter villi and less vasculature, and all of that starts developing because of the tonic signaling provided by microbial stimulation. And another thing that's besides the gut maturing in its absorptive function, you see major changes in immune development. So germ-free mice, they don't have normal lymphoid tissue. If you take a lymph node of a germ-free mouse, it can hardly distinguish where the T cells and B cell areas are. The lymph nodes are tiny. You don't find things like IgA, antibody, at least orders of magnitudes less of that than once you conventionalize them, once you deliver microbes. So this figure shows on the right-hand side is some of the features of the adaptive immune system. So we're looking at T cells and B cells. And the left side column over here, that's germ-free mouse. You don't see many brown dots here. That's, in this case, it's IgA. Here we're looking at a Peyer's patch, so that's a little lymph node-like structure in the intestine. And then CD4 cells, CD8 cells. And you see basically paucity of signal, and all of this is greatly increased after colonization with bacteria. On the right-hand side, you see some of the features of the innate immune system, and the gut has a lot of innate immune mechanisms. Here we're looking at concentrations or expression of some antimicrobial peptides like Regi3, which are induced in the small intestine, including Paneth cells. So basically this is wild-type animal, and you see a lot of red signal, and it does a little bit more detail what kind of signaling is involved, but that's not the main point of this talk. So another idea is the role of microbes as a metabolic organ. So you might expect certain chemicals in circulating in your blood are of microbial origin. In fact, that's about 10%. So one major metabolic organ is liver, but the gut is at least number 2 from the metabolism, what the microbes do. And so this was just a comparison of germ-free animals with conventional mice and looking at various chemicals, what could be microbially derived. And there are some that are absolutely absent in germ-free animals. There are some that are present in much smaller quantities. I'll return to at least one of these later in the talk here, P. cresol, which is a tyrosine metabolite that we kind of stumbled into in our own experiments. But this does raise, a lot of these are neurotransmitter-like, and it potentially could play a lot of different roles. We simply don't appreciate what the microbes are doing and how that could be changed with antibiotics and such. Of course, we always knew microbes were there, but hardly anybody was studying it, and whoever was almost on the fringes of science because there was not much technology to do that. And that changed in the last decade. So we have now ways to look at the microbial world using culture-independent techniques. So in the preceding centuries, the standard was that you had to isolate a specific organism, cultivate it, infect animals that don't have it, see if you can reproduce disease. So basically, I'm going through Koch's postulates. But critical step is that you had to be able to cultivate the organism in the laboratory. This is essentially not possible with the majority of gut organisms because we don't know of all the hundreds, what their particular culture conditions are. They're anaerobes. Many of them prefer to live as communities rather than single organisms. They're interdependent. They're specialized functions. So you have to grow a community, which becomes quite complicated. But anyway, it is possible now just simply to look at their DNA and separate it out on beads or whatever you're using as a technology to separate different DNA strands and sequence everything. And then, of course, you need computational techniques to crunch through these massive amounts of data to come up with some sort of who is there kind of question. And essentially through, I think, my still mid-career, I've seen a birth of a new science, and there are a bunch of terms that still need to be introduced, at least for some members of the audience. A lot of them have meta words, and people throw around different words interchangeably, so I thought I'd just go over the vocabulary. So metagenomics is basically a culture-independent studies of structures and functions of microbial communities. That's another good word, microbial community, or microbial community structure, that you use sequencing of DNA directly from whatever environment you're studying. You could be studying a toilet seat or something else, but it's still metagenomics. Metatranscriptomics, as you might imagine, is you're going a next level, seeing what genes are being transcribed. And then another level is what proteins are being made. But still, you're looking at what the entire community is producing. Microbiota is a term that describes the entire microbial community, actual collection of organisms, that is often switched around with the word microbiome, which is an aggregate of genomes. That refers to the DNA part of it. So microbiome is not necessarily alive, that's just sequence, while microbiota, that's a community of organisms. Then there is a different idea what constitutes a species, since we can't really do classic stuff anymore. People came up with terms like operational taxonomic unit, or OTU, which refers to similarity in 16S ribosomal RNA genes. So all bacteria have these 16S RNA genes, like we have 18S. And you look at sequence similarity because some parts are conserved, but some parts are more variable. And so 97% is similarity is considered a species level. And then you can walk back to lesser degree similarity to genus, family, etc. And even though you might see a complex collection of taxa in your assembly, and everybody in this room has their own fingerprint of what particular species they reside in their gut, when you look at what genes are expressed, functional properties of these different communities, there's a lot more similarity. So the core gene set may be very similar, even though one person has species A doing that function and another person has species B having exact same function in that community. So there's a concept of core genome. So I'm primarily focusing on just two aspects of microbiota: metabolic interactions and immune function, and those are not the only things, but even that's too much for this talk. So one idea is simply energy harvest. So the way we're designed, we're hindgut fermenters. We have a small bowel, and that's where most nutrients get absorbed. But 5 to 10% of energy still comes from microbial metabolism in humans. And that's primarily the fermentation of undigested polysaccharides. So this is kind of a bioreactor. Stuff comes down from small bowel. There's some spillage over of undigested polysaccharides, and those are transformed into short-chain fatty acids like propionate, butyrate, acetate. Butyrate actually is the primary fuel for the colon lining itself. Propionate goes to liver, acetate all over the place. And we get about 5 to 10% of our energy from this. And as I said, we are hind gut fermenters. The fact that we get some energy from bacterial metabolism, we don't normally think about it, but it's not a surprise if you're considering a foregut fermenter like cow, where that's 95% of energy comes from that. And the microbial organ is put up front, where these complex carbohydrates are digested first, and then goes through the complicated intestines. Horse has a complex colon, which I'm happy we don't have to do screening colonoscopies on, but they have large colon and small colon, and the large colon is where the fermentation takes place. And it's some of our close cousins, and you can see they're sitting around, they're eating grass, they have big bellies, they're farting all the time. They're basically fermentation chambers. They have bigger guts than we do, and it's been proposed that a key step in human evolution was cooking. And that's what allowed us to get smaller intestines and bigger brains, so we could occupy ourselves with talks like this and not having to just ferment grass and chew all the time. But still, regardless of that, we have 5 to 10% of energy, and the crosstalk between those bacteria, microbes, and the rest of the body has not gone away. So this was an experiment that turned many heads, including mine, that for some reason it's cutting out the attribution of authorship. This, like half the literature in this field, is coming from Jeff Gordon's lab. A lot of the first authors are independent investigators now. This is Turnbaugh. And so this was a remarkable experiment where they went back to those germ-free animals that are small and colonized them either from normal mice, bacteria taken from normal mice, or ones from genetically obese mice that are here. They're lacking the OB gene. So there's huge fat blobs. And even though the germ-free mice obviously don't have that genetic defect, if you take the microbiota from these obese animals, the germ-free animals gain more weight, as you can see here, this panel here. Both of them gain weight because germ-free animals are small. But if you give them lean bacteria, they gain less weight than if those come from obese animals. And one reason why that may be so is because these microbiota from the obese animals harvest more energy. So if you look at energy content in the cecum at the beginning of the colon, it's actually a little greater in some of these, but in the stool pellets that come out, those calories are more extracted. And then there was a whole bunch of work as to how that is, and there's no way for me to cover all that. But there has been progress in the field, and at least some things worth mentioning is discovery of short-chain fatty acids are ligands for these G-protein coupled receptors, whose names already changed since I've made the slide not so long ago. One is GPR41, one is GPR43. And here of course, people immediately made knockouts of these things. And then you go to germ-free versions of those knockouts. And here we're looking at GPR41, which senses those short-chain fatty acids, which do come from bacterial metabolism of those carbohydrates. And what you notice is the animals that lack this receptor no longer recapitulate that phenotype. When you conventionalize them with microbes, they do not gain weight. So that could be one important mediator of that phenomenon. And where do you find this receptor? Well, it's actually present in neuroendocrine cells. So here it's marked by co-expression of CCK, which is cholecystokinin, which is made by these neuroendocrine cells. Later in this paper, they show that the intestinal transit time is controlled by this receptor, and so they thought maybe energy harvest correlates with that. But another idea was from a Japanese group. Once again, sorry about there is authorship here. they found the densest concentration of this receptor is actually in the sympathetic ganglia. So this receptor has something to say about the metabolic rate of these animals, not just energy harvest. And that got fairly interesting because there's this phenomenon of when you start fasting, how your metabolic rate starts going down, which defeats the weight loss attempts of so many. And why is that? In fact, the short-chain fatty acids may have something to do with that, and they blunt the metabolic rate because of their interaction with the sympathetic tone. And the same phenomenon happens not just in fasting of anybody who wants to do it, but also in diabetics who develop ketone bodies. So specifically, the same receptor also talks to ketone bodies produced during fasting or diabetes. And the short-chain fatty acids produced by microbes interact with that receptor and blunt that communication. I'm not trying to go into specific details of every figure because it's just basic concepts, and there's more mediators. And I'm just trying to make a point that there actually are mechanisms. While in 2006 this was just a wild phenomenon, there are mechanistic ways to look at it. Another one is GPR43, another receptor, and that one is expressed in fat cells. And by adding these short-chain fatty acids, you can make fat cells grow. And of course, people made knockouts of these. And this is a different name for the GPR43. And if you feed them for a while, the empty circle ones here, they instead of keep gaining weight, you have to feed them high-fat diet here to get to mice to close to 50 grams. But the ones that lack this GPR43 or this FFAR2, same thing, stop losing weight later in life. And so this is more of a direct effect on energy intake and expenditure, as they did some metabolic measurements of why that is. So the food intake was in part controlled by this, but also how the energy was spent, or energy storage. So all I wanted to make as a basic concept is the gut microbiota have at least the potential to affect all aspects of energy metabolism. How much you take in, what do you harvest, how do you spend it, and where does it go in your body. I'm not by any means saying that's the sole control of everything. It's just one of the players. Oh, and so in my little cartoon here, these are some of the mediators. So you might imagine there may be a lean microbiota, an obese microbiota that somehow produce different amounts of short-chain fatty acids and have differential signaling on these receptors. And then there are the familiar pathways of metabolic syndrome associated low degree of inflammation and downstream signaling events. Of course, getting fat is not really a health problem by itself. It's the consequences of it, like atherosclerosis. And here again, you can go the next level and see whether microbes can play some role there. And so there's a little body of literature looking at metabolism of phosphatidylcholine, which comes in many dietary elements like cheese, eggs, seafood. And you see the interplay of two metabolic organs, liver and gut, to make different metabolites. So this particular step, choline to trimethylamine, is mediated by gut microbes. And then this trimethylamine, which is a gas, and this is the stuff that makes kidney failure patients smell, that gets metabolized by liver into trimethylamine oxide, and that's an atherosclerogenic compound. And of course, depending on what phenotype you have in the liver, what kind of enzymes you have, may drive differential expression of this step. And there may be differences here. And in fact, people have looked. It's a combination of human and mouse studies. And in the model, for example, ApoE knockout mice which develop atherosclerosis, you can put them on antibiotics, you can suppress atherosclerosis development. And you can find some correlative kind of information on what kind of hepatic gene expression you have in animals that may correlate with atherosclerosis as well, and find similar correlations in humans as well. Another way to look at drug metabolism, at contribution of gut bacteria to metabolism is to look at drug metabolism. Just like liver, it participates in processing a lot of chemicals, so do microbes. Some of these, of course, you need the microbes, for example, to activate sulfosalazine. That's kind of trivial for IBD. Others are more interesting. The fact is that even if you know everything about the liver phenotype of a particular individual, you can only predict about 50% of what's going to happen in that individual patient. And that other 50% may be what kind of gut microbiota they have, because that's the other half. Some drugs like tamoxifen, for example, can cause endometrial cancer. But whether it does or it doesn't depends on the gut phenotype, the gut microbe phenotype that you have, not liver. So this is an example of that, and I'm returning to that p-cresol tyrosine metabolite here. This is metabolism of Tylenol or acetaminophen. It can get glucuronidated or sulfonated. And that happens, of course, in the liver. The tyrosine, this box here, the yellow box, this is what happens as a function of gut metabolism, gut microbe metabolism. And you're starting with tyrosine, and you're getting to p-cresol, which then goes to liver, and it can get sulfonated. Because it's the same enzyme that does both acetaminophen and p-cresol sulfonation, those things can actually interact. And if you happen to be a high p-cresol producer, you will have a somewhat different way of handling Tylenol. And people have tested that. So they collected a bunch of volunteers and measured on this axis the ratio of p-cresol to p-cresol sulfate. And then they challenged them with 1 gram of Tylenol. And the only point on this slide that I want to make is that you have lots of dots and they're scattered in different directions, and people are different. And part of that difference is, in fact, what kind of gut microbes they have. So this leads us, in a way, to what we do, or and the group that we assembled, is this condition of multiply recurrent C. difficile infection. Occasionally it happens in kids, which may be a topic of questions here, but C. difficile does prefer older people, and there's a pretty linear, perhaps even exponential curve of this infection, age being a risk factor. And it's become clear that there is this a syndrome of this infection that's multiply recurrent C. difficile infection. So I'll go through this in a little bit of detail. At least the bird's eye view of pathogenesis of this condition is well understood. Once you start drilling down to details, of course, lots of questions are there. But simply put, we all have microbes living in our colon, as I said. And then when you get antibiotics, not surprisingly, this microbial community gets disrupted. And if you happen to be in the wrong place at the wrong time, like a hospital, you pick up these C. difficile spores, you get an infection with C. difficile, which from spores, these spores germinate, they start making toxin. This toxin kills colonocytes, and you get inflammation, and more severe cases, you have the pseudomembranous colitis, with these mountainous eruptions of neutrophils and a lot of clonocyte death. And of course, you treat this infection with antibiotics as well. C. difficile has a lot of antibiotic resistance, but we have at least a handful that it's sensitive to. And then your hope is to return the patient back to the healthy state. Well, for a first-time infection, there is a 20 to 30% chance that actually it's not going to return here, that it's just going to recycle. And you stop the antibiotics, and the infection just comes back. And then if it happens, if you had one recurrence, the chance of subsequent recurrence keeps going up by 20% every time it happens. And after 3-4 times that it happened and you've tried everything, it seems like this is never going to leave. This becomes a perpetual cycle. And that's the multiply recurrent condition. And the solution to this is to restore the normal composition of microbes, because that's the first line of defense, is actually normal microbes protecting you against this pathogen. And the only way to do it really at this time, because we're so ignorant, is to take the whole microbial community from a normal individual and put it in to restore the normal ecology. And in fact, this is a very effective method of doing this. It was first started in humans as early as 1958. It was the first paper from University of Colorado from Dr. Eisman, who was chief of surgery then, and he's still alive. He's 95 years old and just written a book on care for the aged, and he actually answers email. And then sporadically it's been done over decades, but really this has picked up in the last 5 years or so because of exponential growth of this infection. This became an epidemic. C. difficile did not just waste time all this time. It's become more toxic. So the newer strains are more toxigenic. They also sporulate more efficiently. They leave more spores behind. The organism is spreading into the community. Classically, it's been acquired in medical facilities. About half the infections now are community acquired, et cetera. So all of this kind of worked into a perfect storm where I think there's not an adult gastroenterologist that doesn't once in a while run into this problem at this point. And you kind of feel helpless, which is what happened to me when we started doing this. So when we did start doing this, I thought we should do some science on this instead of just putting poop into people, because that wasn't much progress. And this is pretty much what happens. So this is a study of 3 patients in this case. The first bar is the donor. And I could back up a little bit. We've done about 120 of these by this point. And if you're doing any kind of a high volume thing like this, with a procedure as challenging, at least aesthetically and practically, as this, we have to standardize certain things. And there are 2 steps. One is we decided to develop a standard donor program so that patients wouldn't have to bring in some relative or friend or whatever that we would have to screen, and it's all unreimbursed kind of thing because there's no code, there's no billing code for this. So standard donor was one. The other part was we have to freeze the material. You can't just bring the donor every time you need the stuff. And you need a certain degree of inspiration for it anyway, so it doesn't always work, and you're kind of pacing around endoscopy, oh, come on. So we've overcome these problems, so we kind of have a first-generation product where we have a frozen material, so these 3 patients were actually done with that. And here we're looking at DNA, of course, so it doesn't matter if it's frozen or not. They are the same. And as you can see in this individual, there's the dominance by the blue and red bars. So the blue ones are Firmicutes, and that's gram-positive bacteria, like all the Clostridia not just difficile, but all Clostridial strains would be here. And Bacteroidetes are red. They kind of specialize in polysaccharide digestion, but that's not unique. There's overlap. And then the rest are down here. Actually, unidentified is the next biggest group. And you really have to struggle to see proteobacteria. It's green. Proteobacteria is what you normally, or a lot of people normally think of as gut bacteria. That's like E. coli, Enterobacter, Klebsiella, that group. They're a very minor constituent. They're facultative anaerobes. Typically, they grow a lot better in the culture dish, which is why we get to know them. These other ones are harder to cultivate, and so they're less familiar. But what you see in this second bar is the patient before this fecal transplant, and it is dominated by proteobacteria. I think some of them were like one or two species just took over, like Enterobacter. They have also, this does not reflect it, but there is a lot fewer bacteria. There's orders of magnitude less, because they're being essentially carpet bombed with antibiotics. I don't necessarily think this is a C. difficile induced dysbiosis. This is certainly antibiotic have played a big role in this. And these patients have been on antibiotics for an average in our series of about a year. Just one course after another, and then they get some other infection and have their UTI treated and whatever else. It's never just one antibiotic. So when we have to look at this, all you have to do from a normal is just do a little swab, and you get all the DNA you need. From these people, you need like 5 mils of stool to actually PCR amplify enough DNA. So there is orders of magnitude differences in biomass. And what's, of course, we've observed quite consistently is on day 3, that's our earliest time point, it becomes the donor phenotype. So these bacteria engraft very quickly, and people have usually their first bowel movement on day 3, and this stays fairly stable after that. And we can drill down to lower level of taxa. This is phylum level, just to kind of overview. But it truly does have the fingerprint of the donor bacteria. It's not just there were some remnants that sprang back to life once something normalized. And then there is these black lines here, and that's the levels of p-cresol and p-cresol sulfate. So we thought, as long as we're doing this, and this is the closest we have to a human model of a germ-free something. It's not germ-free, but it's germ-low. Adult human who's getting suddenly reconstituted, then we could look for something that's metabolically different. And p-cresol turned out to be one of those things that's completely undetectable in these patients and quickly comes up following this fecal transplant. Of course, we don't know if it's what exactly it does, if it's good or not or what. Those are different questions. Why it could be interesting because of its interaction with all the sulfonation reactions that happen in the liver, which are important in modulation of hormones and neurotransmitters and chondroitin sulfate, etc. Drug metabolism, higher hyperactivity in children was associated with high p-cresol levels and other neurodevelopmental issues, which I suppose give some credence to the possibility that microbes can play important roles there. Thought I'd touch a little bit on the microbiota immune interactions. Suffice it to say, the immune system does not develop normally at all in absence of gut microbiota, and it's a bilateral conversation. The immune system matures, but the immune system also modulates or shepherds the composition of gut microbes as well. And that's been demonstrated in different mice that lack, for example, IgA or whatever it is. And that could be important as well. This is a somewhat complex slide, but we do have an epidemic of kind of Th2-ish diseases, of allergies and things like that. And there is a concern that use of antibiotics early in life is somehow instructing altered development of your immune system. So this was a rather extreme experiment. They're going with germ-free mice, and they found that they have more of these invariant chain NKT cells compared to normal mice. And this actually development of these cells is an IL-4 dependent event. And there's more eosinophils, and they're kind of Th2-ish. And they're prone to asthma, and which is in the paper as well. And what was, I think, important here is if they normalize the germ-free mice early in life, they could rescue this phenotype and make this Th2-ness disappear. But if they did it to adult germ-free mice, that didn't change. So, I mean, I get a lot of inquiries. Can you fix my allergies with fecal transplant? Well, probably not, but I actually have a scientific rationale why that could be, because it's too late. You already have the problem. So it really goes to a more pediatric age. So there is a developmental aspect to gut microbiota. In fact, it even starts before birth because the pregnant mother has changes in the composition of gut microbes. But then you have a newborn, can go by vaginal delivery or C-section. That's going to influence at least the early colonization, early childhood, the kind of diet you have, antibiotic exposure, etc. And we know the composition of gut microbes in the elderly is different from younger people. We don't know whether it's a cross-sectional, whether it's something they did differently decades ago, and that's why they're different, but they are different. This is looking at what happens in pregnancy. This is an interesting paper in Cell. And what they've shown is that the first trimester composition is different. This is an analytical way to just look at differences between individuals. So first trimester women kind of clustered here. Third trimester clustered a little higher. And this is their database of normal controls. So the pregnant women were different from normal. And then they did a cute experiment is they took the fecal microbiota from these mothers and put them into germ-free animals, and the ones that came from third trimester gained more weight. And they claimed in their paper there's absolutely no dietary difference between first and third trimester and whatever. And they argued that there is a physiologic thirst to gain more weight, and the microbes are helping out, at least in the late stages of pregnancy. However that may be, the baby is born, and this is a study of one baby, most certainly by one of Jeffrey Gordon's postdocs, now a independent investigator. And she followed this baby, nothing can go to waste, for the first 3 years of life, analyzing the diapers all along. And it takes about 3 years for the baby gut microbial composition to mature to an adult-like composition. A lot of things happen here along the course. Introduction of different foods, some hits with antibiotics. This particular baby didn't get a lot of antibiotics, but got some. But because of the increased hygiene and more antibiotics, there's this concern that we may be losing the diversity of our microbiota, and who knows what consequences it has for our health. So in this particular model, there was a thought that we're like losing 20% with every generation, and we may have quite fewer than our great-grandmothers did. Well, there was, of course, when I watched this movie, that's all I could think about, is what's happening to the gut microbes in this case. And so actually the kind of experiment has been done to address this. So people looked at the diversity of microbial community in different geographic locations. So they went to the Amazon and found some Native Americans living in the jungle. They were looking for primitive communities, and as well as to Malawi in Africa, and then 5 cities in the United States, and looked at different ages. And interestingly, actually, for both the more primitive communities and ours, or I should say ancestral communities, I don't think they're necessarily primitive, it takes about 3 years. So this part of the curve has the same slope, and it takes just as long. But what's a little different is where they plateau. So the American subjects plateaued out at about 25-30% fewer species than those in these ancestral communities. Of course, their diet was very different as well. And if you look at the kind of genes that are expressed by these microbes, the Americans look like carnivore animals, and the others look like vegetarians. They had a lot more genes to digest polysaccharides. And that brings to the all these worry about antibiotics in our food supply. Of course, there's been known for a long time that antibiotics will make your farm animals grow bigger and make farming more efficient. And from the very beginning, they knew they were not just treating sickness with that. So here's a quote. So there was unexpected increase in growth rate even when the animals were asymptomatic. And this is subclinical antibiotic dosage, 1953. And now, of course, it's been done in mice, which is far more important. And you can look at more details. And what you see is there's some shifts in the composition. The total number of microbes is the same, but the composition is a little altered. And not that you would care about the details of this, but these are the control mice, and these are this subtherapeutic antibiotic-treated mice. This looks blue, this looks red, because they're looking at difference in gene expression pertaining to metabolism, particularly deposition of fatty acids in the liver. But this was actually done in humans as well, around the same time as the animal farming experiments. So the Navy decided probably that it was not a bad idea to make bigger soldiers, and whether you could do it with antibiotics. So it looks like a pretty decent study. There's about 310 recruits, and they divided them into group 1 that was getting these antibiotics, and then they had a identical control, placebo. And they measured things like sick calls, complaints, hospitalizations, and there were no differences. These soldiers were, surprisingly, by today's standards, fairly small in weight. They're like 150-pound soldiers back in 4. That was one interesting thing. But actually, they did have a positive effect. Even if they started fairly late in their development at age 18, whatever that was, they did get a statistically significant shift in weight in the antibiotic-treated, subtherapeutic antibiotic-treated group. Oh, they didn't look at that. And I don't think they continued that line of development because the society took care of it anyway. So parting thoughts is before 20th century or during most of the 20th century, we looked at microbes from the standpoint of infectious disease, and that is the way our history was. If you just look at George Washington, for example, he had all these things: diphtheria, TB, smallpox, dysentery. He ended up dying from epiglottitis. But we don't, antibiotic. I don't know why I threw that slide in. Oh well. This actually shows. Sorry, this was not meant to be here. But this is endocarditis patients, and they were treated with different antibiotics. Some got amoxicillin, some got vancomycin. or other stuff. And then they looked a year later and noted that the ones that got vancomycin for the same condition ended up gaining more weight. So it's things like you don't necessarily think of looking that could make a difference. And so these days we don't necessarily view some of those infections as the primary problems in our society, but we have plenty problems that may in some way relate to what we're doing with microbes, and our success in conquering some of the infectious disease may have spilled over into whole epidemics of different diseases. There are these associations with allergic disease, asthma, with inflammatory bowel disease. This was a kind of a dose-response curve. The more antibiotics kids got in the first year of their life, the more chance that they were developing Crohn's disease at age 15. And that was replicated across continents. So this is the range of diseases we have now, which may have something to do with microbes that live within us. There was supposed to be an acknowledgement slide, but there isn't. And the people to be acknowledged is we have a team that studies these things, and that includes Mike Sadowski in St. Paul, who is in Biotechnology Institute, and his Department of Soil. And this work kind of brought very unlikely collaborations where you find colleagues that you never thought you even had. And then we have an MD PhD student, Alexa Weingarten, who's doing a lot of this work on fecal transplants, and Matt Hamilton, whose work you may have heard on public radio 2 days ago. He was the one who was blending the poop, and he had some sounds of that. And Chi Chen in nutrition is helping us with the metabolomics. That was a recommendation of Dr. Gorley here, and I think that's working out well. That's all I have. Yes?
I was curious about the process of changing the microbiome after a course of antibiotics. So if you took somebody and put them on a course of antibiotics and sent them to Malawi, would their microbiome look more like that, or would it look like the people they're around, or is it whatever they were beforehand? Are they just repopulating with what they had?
Well, that experiment hasn't been done. What's been done is just taking people, normal volunteers, putting them on ciprofloxacin, and watching how long does it take for the composition to recover back to normal. And that took approximately a month where it looked pretty normal. A certain taxa disappeared forever. So it's possible with every hit of antibiotics you could be losing some diversity. With babies, maybe that's the closest one. At first, if you look, you find mostly mother's bacteria. Depends if they're vaginal or C-section, which ones they are. But after like 3 months and later, they start picking up their own from the environment and building their own community. And so by the time they're done, if you compare to the mother, it's not necessarily that much closer than other family members. Yeah.
Role of probiotics.
Well, my opinion on probiotics is that they're, well, let's see, they're a different topic.
So given that we don't have access to fecal transplants, given that
I view fecal transplant as completely different from probiotics.
right, no,
The idea may be the same from a marketing standpoint, but it's very different because the probiotics really are not mostly adapted to live in the human gut. So they don't really engraft. So you're, as opposed to these do engraft, and they become the new microbiota. I'm very skeptical that the probiotics can do much in the colon. They're like a drop in the ocean by concentration. But there is good data that when you take probiotics, there is change in gene expression in the small intestine. So there, the probiotics do achieve some tickling that is noted by the intestinal tract, and so it's possible that they do something. Whether it's something that's positive or negative, it's impossible to tell, but at least there's a potential that they could do something there. And there is variability in response. Some people do benefit with IBS from probiotics, and some totally don't. And every probiotic is different. It's very hard to study, and of course the makers are not that interested in studying in that detail, and they don't want to make too many health claims, etc. So it's a different area to talk about, but in a common provocative question. Well, the study, the where the studies are. So this has been a different paradigm of how to treat stuff for pretty much everybody involved in regulatory circuits. So our IRB, and I think it's comparable to any other IRB at academic institutions, says if you don't have an IND from the FDA, you cannot conduct a clinical trial. So first you have to go through the FDA. At the moment where it is in FDA, actually FDA has thought about this, and we have an IND to do it from them, but it was a big process. When we had a brief talk with Dr. Eisman right back from the 1950s, he said, well, you had a good idea, you did it back then. That's not exactly the same way. The IND application, I had it weighed. It weighed 22 pounds, just of paper weight. So there's a lot of work to submit it. Anyway, that's kind of where it is. The earliest approvals have just happened. So the IRB approvals just for C. diff are just starting in different places now. So I'm not aware of anybody actually having initiated a study for IBD. And that's what you would have to do. For C. diff, it's a formality because it's unusual in this situation that you have a treatment that's been around for over 50 years, and there is 500 or so patients in various case series that are published with astonishing success rates. So the FDA actually said you can go directly to phase 2. You can skip the phase 1 out of this, but that's unusual. And you have something that's marked improvement over a couple days of these people who've been sick for a year. So the readout is simple. With IBD, it's much more complex, and you absolutely have to have controls. So it's real clinical studies. You can't just glean it from an anecdote. So I'm not aware of any formal studies having been initiated. However, because this has been in the news and stuff, there are some practitioners that are starting to do it, and that was, I guess, expected. There's like a website in Oregon where you can come in for their special resort and for a couple days and get some fecal infusions for whatever price, and I guess they're not being policed. I don't think that's appropriate at all for IBD, because if you're going to do something like that with IBD, you better follow this patient. You might induce a flare. You're the one who's going to be trying to lead this through. Anyway, in our series now, we've had IBD patients with C. diff. And so you could ask, did their IBD go away after this? And there were no real miracles happened that I can say there. If Crohn's disease that lives in the small bowel, I didn't see any change in that at all. I think most of the ulcerative colitis patients did get better, but you could say they got better because we got rid of their C. diff. There are maybe a couple anecdotes where things got dramatically better, but with IBD you're going to have anecdotes, so you really need to do controlled studies. And I actually did a power calculation for this. You need at least 20 patients to do it if the efficacy of this is comparable to Remicade, for example, or infliximab. So there is a paper by Dr. Barodi that everybody quotes on 5 patients that had miraculous cures. There's no denominator. He says he's done about 2,000 overall. He transplants anything that walks into his office. He's in Australia. It's a little different. And so, but that's published, and so patients find that paper and then they ask questions. Yeah.
It's actually been done in humans with different kinds of diets. So, like people who go on a super low-fat diet or the ketogenic diet, things like that. Are those causing permanent changes to the microbiome, or does it sort of give you a shift during that time that you're doing your exclusionary diet? Or do people even know?
I haven't seen such, that those are good studies to do. For example, I know that we use FODMAP diet for IBS quite a bit now. And I know that a study is only now, just one, been published on that particular diet. But I'm not aware of these kind of more extreme diet that would be interesting to do. But that's what...
Based on microbiome in bariatric surgery.
Yes. Mm-hmm. Right. So before and after bariatric surgery, in a handful of patients, and there are some big changes that happen. They're not surprising because you also have an anatomic change and different intestinal transit time and variables like that. But it's a new science, so there's a lot of fruit hanging that you could go after.